Internal structures of plant parts. All living things are composed of one or more cells. In multicellular organism, cells form tissues and various tissues join to form organs. In plants, organs are known as parts. We have studied that plants have two types of tissues, meristematic tissue and permanent tissue. Meristematic tissue comprises dividing cells and permanent tissue consists of various types of cells. These cells are like parenchymatous, collenchymatous, sclerenchymatous, xylem and phloem, etc. Combination of one or two types of cells form three types of tissue systems: epidermal, vascular, ground tissues, each having meristematic cells in it. In this module, you will study how all these cells are arranged in different patterns to form different parts of plant. A plant body is differentiated into root system and shoot system. The shoot system consists of stem, leaves, flowers and fruits, whereas root system comprises several branches of various thickness which help in absorbing water and minerals from the soil. The major parts of a plant body, that is root, stem and leaves, have an important role to play in survival and growth of the plant. Let us explore how all the tissues you studied so far participate in forming different parts of a plant. When you see the transverse section of a dicotyledonous root under a microscope, the cells are found to be organized in different layers like epidermis, cortex, endodermis, and vascular cylinder. This is the outermost covering which is formed of parenchymatous cells and is known as epidermis. A few epidermal cells are modified into hair-like outgrowths known as trichomes, also known as root hair. The root hair increases the absorbing surface of the root. Next region is cortex, which is formed of storage parenchyma that remains active in the uptake of water and minerals. Next few layers of cortex usually consist of thin-walled parenchyma cells with numerous intercellular spaces. Endodermis is one cell thick layer that forms a boundary between the cortex and the vascular cylinder. The endodermis facilitates the movement of water from cortex to inner tissues. Pericycle is a single-celled layer made up of thick-walled, tightly packed parenchyma or sclerenchyma cells that lies just inside the endodermis. Vascular strand lies next to the pericycle, consisting of separate bundles of xylem and phloem arranged alternately. Both are arranged in such a way that it forms an X-shaped pattern in the very center of the root. This is a cross-section of a monocot root. The arrangement of cells is slightly different in monocot root from that of a dicot root. Epidermis. Dermal tissue provides protection of the root. Cortex is wider in a monocot root as compared to a dicot root. Endodermis. It is one cell thick layer forming a boundary between the cortex and the stele. Pericycle is found just inside of the endodermis. It is multi-layered in monocot and responsible for the formation of lateral roots. Major difference of monocot root from dicot root is with vascular tissue. Vascular bundles formed of xylem and phloem are arranged in a ring form, whereas both these tissues, xylem and phloem, form an X when present in a dicot root. Vascular bundles are more than 6 in number. Pith. Centermost region of root is composed of soft, spongy parenchyma cells. It is absent in case of a dicot root. This is a transverse section of a dicotyledonous stem when seen through a microscope. The cells are found to be organized in different layers like epidermis, cortex, vascular bundles, and pith. The epidermis consists of a single layer of parenchymatous cells which are closely packed. The walls are covered with a thin waterproof layer called the cuticle. Main function of the epidermis is to protect the underlying tissues. Cortex. The region is present inside of the epidermis. It is made up of 3 to 4 layers of collenchymatous cells. Beneath the collenchyma cells, there are a few layers of thin-walled cells, parenchyma. The parenchyma cells make up the bulk of the cortex. Collenchyma serves to strengthen the young stem, whereas parenchyma cells store the synthesized food. This region comprises pericycle, vascular bundles and pith. Collectively, it is known as vascular cylinder. The vascular bundles in a dicotyledonous stem are situated in a ring on the inside of the pericycle of the plant. A mature vascular bundle consists of tissues, xylem and phloem. The phloem is located towards the outside of the bundle and the xylem towards the center. Pith. The pith occupies the large central part of the stem. It consists of thin-walled parenchyma cells with intercellular air spaces. The cells of the pith store water and starch. The importance of classification. Classification is a man-made system for the orderly storage and retrieval of data about living world. All members of the living world have to have a name and preferably an allotted place in the huge spectrum of classification. The data entered while classifying living organisms can be used in several ways. It clearly helps in identifying species, studying and conserving species, targeting conservation efforts. Present in the Bowman's capsules, filtering units of kidney. Wall of glomerulus is made up of simple squamous epithelium, thus making this blood filtration possible. These layers of cells are followed by larger thin-walled parenchymatous cells. Intercellular air spaces are found in the parenchyma. Cortex, endodermis and pericycle are not present in monocot stem, but they are there in dicot stem. The vascular bundles are found scattered throughout the ground tissue, whereas dicot stem have vascular bundles forming a distinct visible ring. Pith in monocot stem is not well differentiated as it is in a dicot stem. Cross-section of a dicot leaf. This is transverse section of a leaf when seen through a microscope. A characteristic feature of leaves is the presence of two epidermal layers, one on upper and the other on the lower surface. The ground tissue that occurs between the two epidermal layers is known as mesophyll. Embedded in the mesophyll are the vascular bundles and commonly form the veins. The outer surface of the leaf has a thin, waxy covering called the cuticle. Directly underneath the cuticle is a layer of cells called the epidermis. The vascular tissue, xylem and phloem, are found within the veins of the leaf. Within the leaf, there is a layer of cells called the mesophyll. Mesophyll can then be divided into two layers, the palisade layer and the spongy layer. Palisade cells are more column-like and lie just under the epidermis. The spongy cells are more loosely packed and lie between the palisade layer and the lower epidermis. The air spaces between the spongy cells allow for gas exchange. Mesophyll cells, both palisade and spongy, are enriched with chloroplasts, and this is where photosynthesis actually occurs. Epidermis also lines the lower area of the leaf, where some epidermal cells are modified into guard cells. Two guard cells are arranged in such a way that they form a porous structure known as stomata. Cross-section of a monocot leaf. Monocot leaf is slightly different from dicot leaf. In case of monocot leaf, uniform cuticle is present on both the sides. Numbers of stomata are also equal on both sides, upper as well as lower epidermis. Mesophyll is not differentiated into palisade and spongy parenchyma. As a monocot leaf has veins that run parallel to one another through the length of the leaf, they are visible in the microscopic view like this. To study epidermal cells in Rio leaf. These are the steps to be followed to carry out the activity. Take a freshly plucked Rio leaf, bend between your thumb and forefinger in upward direction. With the help of a forceps, strip a portion of the lower epidermis. Mount the epidermis in a drop of water. Cover it with a cover slip. And observe under microscope. Identify individual cells and take note of the following visible structures: cytoplasm, cell wall, stoma, guard cells, and nucleus. All cells come from previously existing cells. New cells are formed by the process of cell division. To study that apical meristem is present at the tip of root, you can do a small activity. Take 2 equal-sized onions. Take 2 beakers. Fill them with water. Place the onion bulbs on the beakers filled with water in such a way that the roots remain dipped in water. Allow onions to grow. Observe the growth of the roots. The apical meristem present in the root cap region contains the highest percentage of dividing cells. Take out one bulb and cut its root tips. Put it again in the same position. Observe both setups after 3 to 4 days. Roots of the bulb where tips were removed did not grow further. On the other hand, the roots kept growing in case of the other onion. This shows that the tips of the root has meristematic tissue. If removed, the roots cannot grow further. Epithelial tissue. Generally, animals are said to be made up of these 4 basic types of tissues: epithelial, connective, muscular, and nervous. All of these tissues are found in our body, but epithelial tissue covers the whole surface of the body. It also forms the outer as well as the inner covering for most of our internal organs. Characteristic features of epithelial tissues. The tissue provides a covering to our body by forming upper layer of the skin. It also forms covering of our internal organs, means it lines the cavities formed by the internal organ. Primarily, this tissue serves as a protective covering on body surfaces. The cells of epithelial tissue lie on a sticky membrane known as basement membrane. The basement membrane provides structural support for the epithelium and also binds it to neighboring structures. The epithelial cells lack blood vessels, but they get food and oxygen through basement membrane. The cells remain closely packed. The edge of the epithelial tissue that faces the lumen or the outside is called the apical surface. The cells of epithelial tissue are arranged in one or more layers. Epithelial tissue that occurs on the surfaces on the interior of the body is known as endothelium. Actually, epithelium is the name given to the cells that line a surface. The epithelium keeps on dividing to replace itself by the division of the basal layer of the cells. Histological studies of this tissue shows that epithelium can be classified on the basis of two criteria: the number of cell layers present and the shape of the cells on the top layer. On the basis of the number of cell layers present, this tissue is of two types: simple versus stratified epithelium. If an epithelial sheet is composed of one and only one layer, it is a simple epithelium. If there is more than one layer, then it is known as stratified epithelium. Types of epithelial cells. Generally, one of the three different shapes constitutes this tissue. These shapes can be either squamous, cuboidal, or columnar. Squamous cells are thin and flat with flattened nucleus. The cells whose dimensions are more or less equal in all directions, giving a cube-like shape, are identified as cuboidal cells. These cells have round nucleus in the center of the cell. Third shape can be of column type, where the cells are taller than they are wide. These cells have oval nucleus close to the base of the cell. Classification in context to living things is also very important, since it helps us to understand the living world in a better way. Carolus Linnaeus was a Swedish biologist and is considered as a pioneer in the field of taxonomy. He made an attempt to classify living organisms into two kingdoms, the plant kingdom and the animal kingdom. He divided each of these kingdoms into smaller groups called classes. Each class was split into orders. Each order was divided into genera and each genus into many species. Each of these groups was formed on the basis of certain specific morphological features. He described about 5,000 species of plants in his famous book, Genera Plantarum. Similarly, he listed about 4,300 species of animals. He published his scheme Functions of epithelial tissue. Protection. Epithelial cells present as outer skin protect underlying tissue from mechanical injury, harmful chemicals, invading bacteria, and from excessive loss of water. Sensation. Sensory stimuli penetrate specialized epithelial cells. Specialized epithelial tissue containing sensory nerve endings is found in the skin, which helps us to sense different kinds of stimuli. Secretion. In glands, epithelial tissue is specialized to secrete specific chemical substances such as enzymes, hormones, and lubricating fluids. Absorption. Certain epithelial cells lining the small intestine absorb nutrients from the digested food. Excretion. Sweat is also excreted from the body by epithelial cells in the sweat glands. Diffusion. Simple epithelium promotes the diffusion of gases because they form a thin lining which is ideal for the diffusion of gases. The importance of classification. Classification is a man-made system for the orderly storage and retrieval of data about living world. All members of the living world have to have a name and preferably an allotted place in the huge spectrum of classification. The data entered while classifying living organisms can be used in several ways. It clearly helps in identifying species, studying and conserving species, targeting conservation efforts. Present in the Bowman's capsules, filtering units of kidney. Wall of glomerulus is made up of simple squamous epithelium, thus making this blood filtration possible. Simple cuboidal epithelium. The term cuboidal conveys that the cells are equally wide as they are tall, creating a square profile with a spherical nucleus at its center. Simple cuboidal cells are classified as simple since they consist of only a single layer of cells. Simple cuboidal epithelium lines the surface of urinary tubules in the kidney, salivary ducts, and provides a covering to the ovary. Simple cuboidal epithelium lines the walls of the respiratory bronchiole also. Simple columnar epithelium. Cells present in simple columnar epithelium are longer than they are wide. The cells are elongated and column-shaped. The nuclei are elongated and are usually located near the base of the cells. Columnar epithelium forms the lining of the stomach and intestines. Some columnar cells are specialized for sensory reception, such as in the nose, ears, and the taste buds of the tongue. There are certain organs in our body where columnar epithelium is lined by very fine cilia and thus known as ciliated columnar epithelial cells. They are found mainly in the tracheal and bronchial regions of the pulmonary system and also in the fallopian tubes of the female reproductive system. In some locations, epithelial cells are not placed in a uniform pattern. They usually contain more than one type of cells. Glandular epithelium. When columnar epithelial sheet does not contain all cells of the same type but has some goblet cells too, it is known as glandular epithelium. Goblet cells appear as large clear spaces present among the columnar absorptive cells. The cup shape of goblet cells is filled with mucus droplets that are released into the lumen of the tract. Goblet cells are actually unicellular exocrine glands. Examples include sebaceous glands of the skin and glands in the intestinal lining, and many endocrine glands releasing hormones such as thyroid follicle. Let us revise the epithelial tissue. This tissue exists all over the body, covers surfaces, lines cavities, and even forms glands. The epithelial cells set on a thin, non-cellular, gelatinous basement membrane. The epithelial cells lack blood vessels, but they get food and oxygen through basement membrane. It consists of compactly arranged cells which are arranged in one or more layers. The tissue has capacity to get replaced by showing division of the basal layer of cells. Generally, they are classified in 4 categories based on the shape and type of the cells.